Hey guys, welcome to the Human AI podcast. Today we're gonna complete part 2 of talking about glycolysis, the electron transport chain, glucose homeostasis, and glycogen metabolism. If you haven't watched part 1, it's probably a good idea you do because we're gonna continue from where we left off. As you can see here in the screen, we wrote a few things. As always, I have Grok who's gonna be helping us out to understand these topics. Take everything AI says with a pinch of salt and cross-reference. Okay, let's make a start. I want us to talk a bit about the glycerol 3-phosphate shuttle, Grok.
All right, let's dive into the glycerol 3-phosphate G3P shuttle, keeping it concise. The G3P shuttle transfers electrons from NADH, made in glycolysis in the cytoplasm, to the mitochondrial electron transport chain, ETC, for ATP production. Since NAD can't cross the mitochondrial membrane, here's how it works. In the cytoplasm, NADH reduces dihydroxyacetone phosphate to G3P using cytoplasm G3P dehydrogenase. G3P then hands electrons to FAD in a mitochondrial membrane enzyme, mitochondrial G3P dehydrogenase, forming FADH2. FADH2 feeds electrons into the ETC at complex 2, producing about 1.5 ATP per NADH due to FADH2's lower energy yield.
Okay.
This shuttle is...
And what's going on with the malate aspartate shuttle then?
The malate aspartate shuttle moves NADH electrons from glycolysis in the cytoplasm to the mitochondrial electron transport chain more efficiently than the G3P shuttle. Here's the rundown. Cytoplasm NADH reduces oxaloacetate to malate, which enters the mitochondria. Inside, malate is oxidized back to oxaloacetate, regenerating NADH that feeds complex 1 of the ETC, yielding about 2.5 ATP per NADH. To cycle back, oxaloacetate is converted to aspartate, which exits to the cytoplasm, becoming oxaloacetate again. This shuttle dominates in liver, heart, and kidney. Clear? Want to contrast it further with the G3P shuttle or dive into specifics?
Oh, that makes sense. Okay, let me ask you about pyruvate now. What is its role in metabolism?
Pyruvate is a key metabolic hub formed from glucose and glycolysis. Its role depends on conditions. With oxygen, pyruvate enters mitochondria, where pyruvate dehydrogenase converts it to acetyl-CoA, feeding the citric acid cycle to produce NADH, FADH2, and ATP via oxidative phosphorylation. Without oxygen, pyruvate is reduced to lactate in the cytoplasm, lactic fermentation, regenerating NADN+, to keep glycolysis running. Pyruvate can also be used in gluconeogenesis in the liver.
Wait. Remember, be concise. We talked about this. But, yeah, when does it become the oxaloacetate? When does it become lactate? When does it become acetyl-CoA?
Pyruvate's fate depends on cellular needs and oxygen. It becomes oxaloacetate in the liver during gluconeogenesis using pyruvate carboxylase to make glucose when blood sugar is low. It turns into lactate in the cytoplasm under low oxygen, anaerobic conditions, via lactate dehydrogenase to regenerate NADN+ for glycolysis. It's converted to acetyl-CoA in mitochondria with oxygen by pyruvate dehydrogenase, entering the citric acid cycle for energy production. Clear? Want specifics on any path?
I don't get it.
No problem. Let's simplify. Pyruvate, made from glucose and glycolysis, has 3 fates. 1. Low oxygen, it turns into lactate in the cytoplasm to keep glycolysis going. 2. Need glucose? In the liver, it becomes oxaloacetate to build glucose via gluconeogenesis. 3. Got oxygen? It's converted to acetyl-CoA in mitochondria for the citric acid cycle to make energy. The cell picks based on energy needs and oxygen levels. Clear now? Want to focus on one fate?
Okay. Okay, so when there's sufficient oxygen, we can oxidize pyruvate and we can generate ATP, carbon dioxide, water. In hypoxic conditions, it's reduced to lactate. So we have aerobic conditions in the mitochondria, and then we have glycolysis in the cytosol. Can you explain why one is in the cytosol and the other in the mitochondria?
Glycolysis happens in the cytosol because it's a fast, ancient pathway that doesn't need organelles, breaking glucose into pyruvate to quickly generate ATP and NADH. It's self-contained, using enzymes free in the cytoplasm, ideal for rapid energy in any cell, even without mitochondria. Aerobic pyruvate oxidation occurs in mitochondria because it relies on the citric acid cycle and electron transport chain, which are housed there. Mitochondria have the machinery, enzymes like pyruvate dehydrogenase, membrane-bound ETC complexes, and oxygen access to efficiently convert pyruvate to acetyl-CoA, then CO2, water, and lots of ATP via oxidative phosphorylation. The compartmentalization separates rapid, low-yield glycolysis from high-yield, oxygen-dependent processes. Clear? Want more on the enzymes or ATP yields?
That makes sense. Okay, let's talk a bit more about the transport of pyruvate into the mitochondria then. So this happens in aerobic conditions via a specific carrier protein in the mitochondrial membrane. And then tell us what's going on. We have decarboxylation, oxidative, by the pyruvate dehydrogenase complex to form acetyl-CoA. Explain that.
Pyruvate transport and conversion to acetyl-CoA, keeping it concise. In aerobic conditions, pyruvate crosses the mitochondrial inner membrane via a specific carrier, the mitochondrial pyruvate carrier MPC, a protein that moves pyruvate from the cytosol into the matrix. Once inside, the pyruvate dehydrogenase complex, PDC, a multi-enzyme complex, catalyzes pyruvate's oxidative decarboxylation. It strips a carbon from pyruvate as CO2, oxidizes it using NADN+ to form NADH, and links the remaining 2-carbon unit to coenzyme A, yielding acetyl-CoA. This acetyl-CoA enters the citric acid cycle for ATP production. PDC's action is tightly regulated to match energy needs. Clear? Want details on PDC regulation or the citric acid cycle link?
Makes sense. Let's talk a bit about the TCA cycle.
The TCA cycle, or citric acid cycle, Krebs cycle, is a mitochondrial pathway that oxidizes acetyl-CoA to produce energy carriers. Acetyl-CoA, from pyruvate or fatty acids, combines with oxaloacetate to form citrate. Through a series of enzyme-driven steps, citrate is reshaped, releasing 2 CO2, regenerating oxaloacetate, and producing 3 NADH, 1 FADH2, and 1 GTP, similar to ATP per cycle. NADH and FADH2 feed the electron transport chain for ATP synthesis. It's a central hub, linking glycolysis, fat, and protein metabolism to energy production. Clear? Want specifics on TCA enzymes or its regulation?
I don't get it.
No worries. Let's simplify. The TCA cycle in mitochondria takes acetyl-CoA from pyruvate and mixes it with oxaloacetate to make citrate. It then reshapes citrate through steps, spitting out 2 CO2, 1 GTP like ATP, 3 NADH, and 1 FADH2 per round, while remaking oxaloacetate. NADH and FADH2 fuel the electron transport chain to make lots of ATP. It's the cell's main energy factory.
So acetyl residues are oxidized to CO2.
Exactly. In the TCA cycle, the acetyl group from acetyl-CoA, 2 carbons, is oxidized, releasing 2 CO2 molecules per cycle. This happens as enzymes reshape intermediates, with electrons from the acetyl group transferred to NADH plus and FAD, forming NADH and FADH2, which drive ATP production in the electron transport chain. Clear? Want to dive into
So
the
from
oxidation?
acetyl-CoA we go to citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, L-malate, oxaloacetate, and we have all these steps. Okay.
Yep, you've got the TCA cycle intermediates down. Let's make it clear and concise, tying names to what's happening. Acetyl-CoA enters the cycle, merging with oxaloacetate to form citrate. Citric acid, hence the cycle's name. Here's the flow and why each step matters. 1. Citrate to isocitrate. Aconatase. Citrate is rearranged into isocitrate, a minor structural tweak to prep for oxidation. Iso reflects the aconatase. 2. Isocitrate to alpha-ketoglutarate. Isocitrate dehydrogenase. Isocitrate loses a carbon as CO2, gets oxidized, and forms alpha-ketoglutarate, producing NADH, named for its ketone group. 3. Alpha-ketoglutarate to succinyl-CoA. Alpha-ketoglutarate dehydrogenase. Another carbon is lost as CO2, with oxidation yielding NADH and attaching coenzyme A to form succinyl-CoA, named for the substrate. 4. Succinyl-CoA to succinate. Succinyl-CoA synthesis. Succinyl-CoA releases coenzyme A, forming succinate and GTP like ATP. Succinate reflects its 4-carbon structure. 5. Succinate to fumarate. Succinate dehydrogenase. Succinate is oxidized, producing FADH2, and forms fumarate, a double-bonded molecule, named for its structure. 6. Fumarate to L-malate. Fumarase. Fumarate gains a water molecule, forming L-malate. Fumarase comes from its substrate. 7. L-malate to oxaloacetate. Malate dehydrogenase. Malate dehydrogenase. L-malate is oxidized, yielding NADH and regenerating oxaloacetate, ready for another round, named for its malate target.
Wow. Okay. Let's talk a bit about the product then. We have 3 NADH, 1 FADH2, and 1 GTP. What's a GTP?
The TCA cycle produces 3 NADH, 1 FADH2, and 1 GTP per acetyl-CoA. GTP, or guanosine triphosphate, is an energy molecule like ATP, carrying a high-energy phosphate bond. In the cycle, succinyl-CoA synthesized, forms GTP from GDP, an inorganic phosphate, while converting succinyl-CoA to succinate. GTP can directly fuel cellular processes or transfer its phosphate to ADP to make ATP, depending on the cell's needs. NADH and FADH2 carry electrons to the electron transport chain for ATP production. Clear? Want to focus on NADH, FADH2 roles, or GTP's use?
I want to ask about anaplerotic reactions.
All right, let's switch gears to anaphylactic reactions.
No. ANAPLEROTIC has to do with the TCA cycle.
My bad, I misheard. Let's talk anaphylactic reactions in the TCA cycle. Anaphylactic reactions replenish TCS cycle intermediates, like oxaloacetate or alpha-ketoglutarate, when they're pulled out for other processes, like gluconeogenesis or amino acid synthesis. Key examples: pyruvate carboxylase converts pyruvate to oxaloacetate, and glutamate dehydrogenase turns glutamate into alpha-ketoglutarate. These reactions keep the cycle running by refilling its intermediates, ensuring acetyl-CoA can be oxidized for energy. Clear? Want specifics on anaphylactic enzymes or their regulation?
I want to ask you, let's kind of, sorry guys, my housemate wants to throw a ball at me. So let's focus a bit on something different now, about passive and active. Sorry guys, I feel threatened by a squishy ball thrown at me. How unprofessional. Okay, let's talk about the passive and active transport of molecules into cells. And
No word.
Then let's talk about glucose transport into tissues. GLUT 1, 2, 3, 4, 5.
All right, let's hit passive and active transport, then glucose transport into tissues, keeping it concise. Passive transport moves molecules like glucose across cell membranes down their concentration gradient. No energy needed. It uses facilitated diffusion via transporters, like GLUT proteins for glucose. Active transport moves molecules against their gradient, requiring energy, usually ATP or ion gradients. Sodium glucose cotransporters, SGLT, in intestines and kidneys are examples, pumping glucose uphill. Now, glucose transport. GLUT1, brain, red blood cells. GLUT2, liver, pancreas. GLUT3, neurons. GLUT4, muscle, fat, insulin regulated. And GLUT5, intestine,
Can you
fructose.
stop? You're mispronouncing it maybe. It's GLUT, not GLUT. Okay, let's talk a bit about GLUT1. So GLUT1 is found in erythrocytes, muscle, the brain, quite a lot of tissues. GLUT2 is specifically in the liver. GLUT3 is in the brain. GLUT4 is in the skeletal muscle. So basically they're in different locations. Okay. Is there a way to remember which one goes where without memorizing? Is there some kind of rationale behind it?
Yeah, there's a rationale behind why GLUT transporters, like GLUT3 and GLUT4, are in specific tissues so you can understand rather than memorize. Here's a concise breakdown to make it stick, tied to function and tissue needs. GLUT3 is in neurons' brain because they have a high constant glucose demand for energy, and GLUT3 has high affinity, grabs glucose even at low levels, and is always on the membrane. Think, brain's priority is non-stop fuel for thinking. GLUT4 is in muscle and fat because these tissues store energy and need regulated glucose uptake. GLUT4 is insulin dependent, moving to the membrane only when insulin signals high blood sugar, preventing excess uptake otherwise. Think, muscle fat as glucose storage units, activated post-meal. Other GLUTs? GLUT1, red blood cells, brain barriers, is ubiquitous, low-level glucose supply. GLUT2, liver, pancreas, handles high glucose, sensing and managing blood sugar. GLUT5, intestine, is for fructose, not glucose. The logic? Each GLUT matches its tissue's glucose needs. High demand, neurons, GLUT3. Regulated storage, muscle fat, GLUT4. Or sensing, liver, GLUT2. Picture tissues as machines with custom fuel pumps. Clear? Want tips to visualize or more on one GLUT?
Well, I do want to ask you, okay, well, fair enough. Yeah, but how did they name them 1 to 5? Is it chronologically?
The naming of GLUT1 to GLUT5 isn't chronological, but based on the order of their discovery and cloning in the 1980s, 1990s.
Okay, so let's talk about insulin regulation a bit then. How it connects to GLUT4 glucose transporter, PKC, Pi3 kinase.
Insulin regulates glucose uptake by activating GLUT4 through a signaling cascade involving Pi3 kinase and, indirectly, PKC. When insulin binds its receptor on muscle or fat cells, the receptor autophosphorylates, activating Pi3 kinase, phospholinositide 3 kinase. Pi3 kinase generates Pi3 phosphatidylinositol 3,4,5 trisphosphate, which recruits and activates AK-ET, a kinase. AK-ET signals vesicles containing GLUT4 to move to the cell membrane, increasing glucose uptake by facilitated diffusion. Protein kinase C, PKC, can play a supporting role in some cells, amplifying insulin signals or aiding vesicle trafficking, but Pi3 kinase AK-ET is the main driver for GLUT4 translocation. This ensures glucose enters cells only when blood sugar is high, signaled by insulin. Clear? Want more on signaling steps or GLUT4 mechanics?
No, it's okay.
All right, what's next? Got another topic?
So with insulin, we have some longer lasting effects. What that interests me is that you have an increase in expression of adipocyte enzymes that synthesize glycerols, and it also inhibits lipolysis. It's not that bad.
Insulin's longer-lasting effects promote fat storage and block fat breakdown. It increases expression of adipocyte enzymes like lipoprotein lipase and glycerol 3-phosphate dehydrogenase, which helps synthesize triglycerides, not dry sialoglycerols, by providing fatty acids and glycerol 3-phosphate for fat storage in adipose tissue.
So similarly to what it does to glucose, basically.
Exactly. Insulin's role with triglycerides mirrors its glucose effects.
Okay, now, let me ask you about pentose phosphate pathway.
The pentose phosphate pathway, PPP, in the cytosol has two phases and runs parallel to glycolysis. Oxidative phase. Glucose 6-phosphate is oxidized, producing NADPH for biosynthesis and antioxidant defense, and ribulose 5-phosphate with CO2 released. Non-oxidative phase. Ribulose 5-phosphate is converted to ribose 5-phosphate for nucleotide synthesis or other sugars, like fructose 6-phosphate, that can feed back into glycolysis. It's active in tissues like liver, adipose, and red blood cells, balancing NADPH and nucleotide needs with energy metabolism. Clear? Want details on enzymes or its regulation?
How many? So 1,000 minutes. How many hours is that?
1,000 minutes divided by 60 minutes per hour is about 16.67 hours. Clear? Want to jump back to the pentose phosphate pathway something?
All good. Okay.
Cool.
So, okay, let's jump topics again, and let's continue talking about the liver a bit and glucose 1-phosphates going to 6 and all that jazz.
All right, let's focus on the liver and glucose 1-phosphate to glucose 6-phosphate and glycogen metabolism. In the liver, glycogenolysis breaks glycogen into glucose 1-phosphate via glycogen phosphorylase. Phosphoglucomutase then converts glucose 1-phosphate to glucose 6-phosphate. In the liver, glucose 6-phosphate can be further processed by glucose 6-phosphatase to produce free glucose, which is released into the blood to maintain blood sugar, especially during fasting. Alternatively, glucose 6-phosphate can enter glycolysis for energy or the pentose phosphate pathway for NADPH and ribose. This flexibility makes the liver a key glucose regulator. Clear? Want more on liver enzymes or glucose regulation?
Well, I want to ask you about transferase activity of the debranching enzyme, removing 3 residues from the branch and transferring them to the end of another chain in an alpha 1,4 linkage.
The debranching enzyme in glycogenolysis has transferase activity that handles glycogen's alpha 1,6 branch points. When glycogen phosphorylase reaches a branch, it stops, leaving a 4-glucose residue stub linked by an alpha 1,6 bond. The debranching enzyme's glucosylase transferase activity transfers 3 of these glucose residues to the end of another glycogen chain, linking them via an alpha 1,4 bond, extending the linear chain.
To be honest, I don't get it. If you could explain it a bit more, I would be very happy. Like, maybe if you could explain it from a different angle and explain after how the single glucose unit left at the branch is removed by the action of an alpha 1,6 glucosidase activity of the debranching enzyme. I don't know what these things mean either. If you could explain to me really nicely in 2-3 sentences.
Glycogen's branches, linked by alpha 1,6 bonds, are trimmed by the debranching enzyme, which uses its glucosylase transferase to move 3 glucose units from a 4-glucose branch stub to a linear chain, attaching them with an alpha-1,4 bond for further breakdown. The single glucose left at the branch is then removed by the enzyme's alpha-1,6 glucosidase activity, which cleaves the alpha-1,6 bond to release free glucose. This dual action ensures glycogen is fully broken down into glucose units for energy or blood sugar maintenance. Clear?
Okay, I see.
Awesome, glad it's clicking. What's next? Ready to dive into another topic or stick with glycogen?
Well, I want to ask you, so from glucose 1-phosphate, UTP is in between that and uridine diphosphate glucose, UDP glucose, what's going on there?
From glucose 1-phosphate to UDP glucose, the liver builds glycogen precursors. Glucose 1-phosphate reacts with UTP, uridine triphosphate, via UDP glucose pyrophosphorylase, forming UDP glucose and releasing pyrophosphate. UDP glucose then donates glucose units to growing glycogen chains during glycogenesis, catalyzed by glycogen synthase, with UTP providing the energy to link glucose to the uridine carrier. Clear? Want more on glycogenesis or UTP's role?
Well, I don't get it, what this is, to be honest. And how it connects to glycogen synthase too.
No problem. Let's simplify. Glucose 1-phosphate is activated into UDP glucose by the enzyme UDP glucose pyrophosphorylase, which uses UTP, an energy-rich molecule, to attach uridine to glucose 1-phosphate, releasing pyrophosphate. UDP glucose is then the active form that glycogen synthase uses to add glucose units to a growing glycogen chain, forming alpha-1,4 linkages during glycogenesis. Clear? Want to focus on glycogen synthase's action or UTP's energy role?
I see. Okay. Let me ask you about the formation of glycogen from UDP glucose then. So we have glycogen synthase, and then it adds glucose units in alpha-1,4 linkage onto the glycogen chain using the UDP glucose.
Exactly. Glycogen synthase takes UDP glucose and adds its glucose unit to the growing glycogen chain, forming an alpha-1,4 linkage, extending the chain. The UDP is released, and the process repeats to build glycogen during glycogenesis in the liver and muscles. For branching, glycogen branching enzyme later adds alpha-1,6 linkages to create glycogen's tree-like structure. Clear? Want details on branching or regulation?
That's okay. Okay, I want to ask you about, let's go back to the ETC. So we have a flow of electrons and protons through complexes of the ETC. And
All right.
I want to understand the path of the electrons entering the electron transport chain.
The electron transport chain, ETC, in the mitochondrial inner membrane moves electrons from NADH and FADH2 through complexes to generate ATP. NADH donates electrons to complex 1, NADH dihydroxide, NADH2, which passes them via ubiquinone, coenzyme Q, to complex 3, succinate dihydroxide, also via ubiquinone to complex 3. From complex 3, electrons move through cytochrome C to complex 4, cytochrome C oxidase, where they're used to reduce oxygen to water. Electron flow pumps protons, H+ protons, across the membrane, creating a gradient that drives ATP synthase to make ATP. Clear? Want details on proton movement or ATP yield?
Not that clear. No.
No worries, let's simplify. Electrons from NADH enter the ETC at complex 1, flow through ubiquinone to complex 3, then via cytochrome C to complex 4, reducing oxygen to water. FADH2 electrons start at complex 2, joining the same path via ubiquinone. This electron movement powers proton pumping, driving ATP synthase to produce ATP. Clear now? Want to focus on one complex or protons?
I don't get it.
Okay, let's try a different angle. Super simple. Imagine NADH and FADH2 as electron donors. NADH hands electrons to complex 1, FADH2 to complex 2. These electrons zip through ubiquinone, aka U, a courier to complex 3, then cytochrome C, another courier to complex 4, where they combine with oxygen to make water. This electron relay pumps protons to create energy for ATP synthase to make ATP. Clear? Want to zoom in on any step?
Yeah, that kind of makes sense. Okay. And let's go forward with that. So let's talk a bit about synthesis of ATP. So we've transported ADP and PI into the matrix and synthesize ATP and transport it to the cytosol, right? Okay, fair enough. Since the matrix is kind of has all the tools, it's like giving my broken phone to the place and then bringing it back home, I guess, or something.
Haha, love the broken phone analogy. Let's clarify ATP synthesis and transport, keeping it concise. In the mitochondrial matrix, ATP synthase uses the proton gradient from the electron transport chain, ETC, to combine ADP and inorganic phosphate, Pi, into ATP. The ETC pumps protons out, and their flow back into the matrix through ATP synthase spins it to make ATP. ADP and Pi are transported into the matrix via specific carriers, like the adenine nucleotide translocase for ADP and phosphate translocase for Pi. Once made, ATP is shuttled back to the cytosol by the same translocase, exchanging ATP for ADP.
Explain me a bit more this adenine nucleotide translocase, known as the anti-porter.
The adenine nucleotide translocase, ANT, also called the ADP-ATP antiporter, is a protein in the mitochondrial inner membrane that swaps ATP and ADP. It transports ATP made in the matrix by ATP synthase to the cytosol for cellular use, while bringing ADP back into the matrix to be reused for more ATP synthesis. This antiporter works by exchanging one ATP, 3 negative charges, for one ADP, 2 negative charges.
And what's phosphate translocase?
The phosphate translocase is a protein in the mitochondrial inner membrane that transports inorganic phosphate, Pi, into the matrix. It works as a symporter, moving Pi alongside a proton, H plus proton, into the matrix, driven by the proton gradient created by the electron transport chain. This ensures Pi is available for ATP synthase to combine with ADP to form ATP during oxidative phosphorylation. Clear? Want to tie this back to ATP synthesis or another topic?
I don't get phosphate translocase, to be honest, and I would also like to understand what symporter means.
No problem. Let's break it down simply. The phosphate translocase is a mitochondrial membrane protein that moves inorganic phosphate, Pi, into the matrix, where it's needed for ATP synthase to make ATP from ADP and Pi. It's a symporter, meaning it transports 2 molecules in the same direction. Here, Pi and a proton, H plus proton, using the proton gradient's energy to pull Pi into the matrix. Think of it as a gate shuttling pi with a proton buddy to fuel
I don't
ATP.
get it. I don't get why we need it. Why we don't stop with the previous one we talked about.
Alright, let's clear up why we need the phosphate translocase alongside the adenine nucleotide translocase, ANT. The ANT swaps ATP out of the matrix for ADP coming in, handling the nucleotide part of ATP synthesis. But ATP synthase also needs inorganic phosphate, pi, to combine with ADP to make ATP. The phosphate translocase is essential because it brings pi into the matrix, using the proton gradient to move pi with a proton as a symporter.
But I thought that already ATP came in, no?
I see the confusion. Let's straighten it out. The adenine nucleotide translocase, ANT, doesn't bring ATP into the matrix. It exports ATP made in the matrix to the cytosol and imports ADP from the cytosol into the matrix. ATP synthase in the matrix needs both ADP and inorganic phosphate, pi, to make ATP. The phosphate translocase supplies the pi by moving it into the matrix with a proton, using the proton gradient. So, ANT handles ADP-ATP exchange, while phosphate translocase delivers pi. Both are needed to keep ATP production going. Clear now?
No.
Want to Alright, cool. What's next?
No, I
Want
said
to
no, I don't get it.
My bad. I misunderstood. Let's try again. Super simple. ATP synthase in the mitochondria needs two things to make ATP: ADP and phosphate, pi. The adenine nucleotide translocase, ANT, brings ADP into the matrix and sends finished ATP out to the cell. But pi, the other ingredient...
Pi will come with phosphate translocase then.
Exactly. The phosphate
And why one is called antiporter and the other symporter? Tell us this, and then we'll talk a bit about ATP synthase. Tell us why it's F-type and what's the difference between F0 and F1.
The adenine nucleotide translocase is an antiporter because it swaps molecules in opposite directions: ADP into the matrix, ATP out. The phosphate translocase is a symporter because it moves two molecules in the same direction, pi and a proton, into the matrix. Now, ATP synthase, an F-type ATPase, synthesizes ATP using the proton gradient. It's called F-type due to its fraction designation from early studies, distinguishing it from other ATPases, like V-type or P-type. ATP synthase has two parts. F0, embedded in the mitochondrial membrane, forms a proton channel, letting protons flow in. F1, in the matrix, binds ADP and pi, using the proton-driven rotation of F0 to catalyze ATP formation. Clear? Want more on ATP synthase mechanics or its regulation?
I don't get it.
No worries. Let's simplify. An antiporter, adenine nucleotide translocase, trades ADP in for ATP out, like a two-way swap. A symporter, phosphate translocase, pulls pi and a proton into the matrix together, same direction. ATP synthase, called F-type for historical naming, makes ATP with two parts. F0, a membrane channel letting protons flow in, spins F1, a matrix component that uses that spin to stick ADP and pi together into ATP. Clear now? Want to focus on one part, like F0, F1, or porter types?
I don't get this F0, F1 thing. So F0 has three different types of subunits. F1 comprises 5. I don't get the naming though. The reasoning.
Okay, let's clear up the F0 and F1 naming and structure for ATP synthase, keeping it simple. The F-type ATP synthase is named F from fraction in early biochemical studies, not for function. F0 and F1 are its two main parts, labeled for their discovery as distinct fractions. F0, 0, is the membrane-embedded part, with 3 subunit types, A, B, C, and mammals, forming a proton channel and rotor. The 0 reflects its initial identification as a base fraction. F1, 1, is the matrix exposed part, with 5 subunit types, alpha, beta, gamma, delta, etel, where beta subunits catalyze ATP synthesis using rotation driven by F0. The 1 just follows as the next fraction identified. The names are historical, not descriptive, but F0 moves protons. F1 makes ATP. Clear? Want to dive into subunit roles?
I don't get it, I can't lie. And why do the 3 beta subunits have catalytic sites for ATP synthase?
No problem. Let's make F0 and F1 super clear and tackle the beta subunits. ATP synthase F0 and F1 names are just old lab labels from when scientists split it into parts. F0, membrane part, and F1, matrix part. Think of F0 as the engine's base, letting protons flow, and F1 as the factory making ATP. F0 has 3 subunit types, A, B, C, forming a spinning proton channel. F1 has 5 subunit types, alpha, beta, gamma, delta, etel, and the 3 beta subunits are the stars because they hold catalytic sites where ADP and pi combine to form ATP. Why beta? Each beta subunit is ideally positioned in F1 structure.
Wow, this is really hard stuff. And the gamma unit rotates, and then the beta catalytic unit, beta catalytic units properties change. How does that happen?
Yeah, ATP synthase is tricky, but let's break down the gamma unit rotation and beta subunit changes simply. In F1 of ATP synthase,
yeah, continue. Why stop?
Alright, APK makes it so because its spinning mechanism is like a molecular machine, but let's keep it clear. The gamma Gemini subunit in F1 rotates, driven by proton movement through F0 C subunits. This rotation pushes against the 3 beta subunits, which are fixed in a ring around gamma. Each beta subunit's catalytic cycles through 3 shapes, open, loose, tight, as gamma turns, like a crank shifting gears. In the open shape, ATP and pi enter. Loose holds them, tight squeezes them into ATP, which is released as the cycle repeats. This conformational change driven by gamma's rotation ensures constant ATP production. The complexity comes from visualizing this dynamic, multi-subunit dance powered by protons. Clear?
No, no, that's all. I also don't understand about the electrochemical energy generated in the intermembrane space as the difference in charge and difference in proton concentration.
No worries. Let's simplify the electrochemical energy and ATP synthesis role. The electrochemical...
I also want to understand the previous thing we were talking about. I said not at all. I said no, I don't understand that either.
My bad. Let's backtrack and clarify both the ATP synthesis beta subunit cycle and the electrochemical energy, keeping it super simple. First, ATP synthesis. The gamma Gemini subunit in F1 spins, powered by protons moving through F0. This spin pushes on 3 beta subunits, which have catalytic sites. As gamma turns, each beta site changes shape, like a door opening closing, and locking open lets ADP and pi in. Loose holds them, tight forms ATP, then opens to release it. This cycle repeats making ATP. Now the electrochemical energy. The electron transport chain, ETC, pumps protons, H-pi, from the matrix to the intermembrane space, creating a gradient. More protons outside, higher concentration, and a charge difference outside more positive. This proton motive force drives protons back into the matrix through F0, spinning gamma to power ATP synthase. Think of it as water pressure turning a turbine. Clear? Want to focus on one part or try another angle?
Kind of clear. Okay, let's jump to our previous topic. Let's talk a bit about glycogen synthesis then. So, glycogen synthase as the glucose units of more than 4 glucosyl residues. And then what does glycogenin do? Glycogenin, tyrosine and UDP glucose do?
All right, let's dive into glycogen synthesis, focusing on glycogen synthase, glycogen, tyrosine, and UDP glucose, keeping it concise. Glycogen synthase adds glucose units from UDP glucose to a glycogen chain, forming alpha-1,4 linkages, but it needs a primer to start. Glycogen is that primer, a protein that self-glucosylates, attaching UDP glucose to its own tyrosine residue, an amino acid side chain to form a short glucose chain, about 8-12 units. This chain serves as the base for glycogen synthase to extend into a full glycogen molecule. UDP glucose provides the activated glucose units, releasing UDP as each glucose is added. Clear? Want more on glycogen's role or branching enzyme?
I don't get it at all, in fact.
No problem. Let's make glycogen synthesis crystal clear, step by step. Glycogen synthase can't start a glycogen chain from scratch, so it needs a starting point. Glycogen, a special protein, kicks things off by attaching a glucose from UDP glucose, an activated glucose molecule, to a tyrosine, an amino acid on itself, like sticking a sugar to its own arm. Glycogen keeps adding a few more glucoses from UDP glucose to make a short chain. Then, glycogen synthase takes over, using UDP glucose to add more glucoses to this chain, linking them with alpha-1,4 bonds to build the glycogen molecule. UDP glucose is the glucose donor, releasing UDP each time a glucose is added. Think of glycogen as the seed, and synthase as the gardener growing the glycogen tree. Clear now? Want to focus on one part, like UDP glucose?
I don't get it, bro.
Alright, bro, let's make this glycogen synthesis thing dead simple, like explaining it over a coffee. Glycogen's a big sugar storage molecule, but building it needs a starting block. Glycogen, a protein, acts like a tiny anchor. It grabs a glucose from UDP glucose, a ready-to-go glucose package, and sticks it onto its own tyrosine, an amino acid hook on glycogen. This creates a small glucose chain, like a seed. Then, glycogen synthase jumps in, piling on more glucoses from UDP glucose, linking them with alpha-1,4 bonds to grow the glycogen molecule. UDP glucose is just the fuel, spitting out UDP each time it donates a glucose. So, glycogen...
that makes more sense. Okay, so let's talk a bit more about glycogen as an energy store. So, why is the branch structure a bad store? Something to do with being hydrophilic.
Glycogen's branch structure makes it an excellent energy store, not a bad one, and its hydrophilic nature is key. The alpha-1,4 and alpha-1,6 linkages create a tree-like branched molecule, allowing rapid breakdown because enzymes like glycogen phosphorylase can attack multiple branch ends at once, quickly releasing glucose 1-phosphate for energy. Being hydrophilic, water-attracting, glycogen binds water, staying soluble in the cytosol of liver and muscle cells.
But doesn't it associate with water increasing the overall weight and bulk?
You're right. Glycogen's hydrophilic nature means it binds water, which does increase its weight and bulk, about 2-4 grams of water per gram of glycogen in cells. This might seem like a downside for storage efficiency, but it's a trade-off that makes glycogen a great energy store. The water keeps glycogen soluble and accessible in the cytosol, allowing enzymes like glycogen phosphorylase to quickly break its branch structure, alpha-1,4 and alpha-1,6 linkages, into glucose 1-phosphate for fast energy release, especially in liver and muscle.
Okay.
Linear...
So, can I ask about the allosteric regulation of phosphorylase?
Glycogen phosphorylase, which breaks glycogen into glucose 1-phosphate, is tightly controlled by allosteric regulation to match energy needs. In muscle, phosphorylase exists as active phosphorylated, called phosphorylase A, and less active, dephosphorylated, phosphorylase B forms. Allosteric activators, like AMP, high when ATP is low, boost phosphorylase B activity, signaling energy demand, while ATP and glucose 6-phosphate inhibit it, indicating energy sufficiency. In the liver, glucose acts as an allosteric inhibitor, slowing phosphorylase when blood sugar is high. Calcium, during muscle contraction, and glucagon epinephrine, via phosphorylation, also activate phosphorylase for quick glucose release. This fine-tunes glycogen breakdown. Clear? Want details on hormonal control or specific effectors?
I don't get this allosteric regulation of phosphorylase.
No worries. Let's simplify allosteric regulation of glycogen phosphorylase. It's an enzyme that breaks glycogen into glucose 1-phosphate.
I also don't understand allosteric what it means. So tell me what it means too, and then tell me how it relates to AMP that activates phosphorylase. Why is AMP the one? And yeah.
All right, let's break this down clearly and concisely. Allosteric regulation means a molecule binds to an enzyme at a site other than its active site, changing its shape and activity, like a key tweaking a lock's function. For glycogen phosphorylase, which breaks glycogen into glucose 1-phosphate, AMP, adenosine monophosphate, is an allosteric activator, especially in muscle. When energy is low, AMP levels rise, binding phosphorylase's allosteric site, boosting its activity to release more glucose for ATP production. Why AMP? It signals low energy since ATP breaks down to AMP when energy is scarce, making it a perfect trigger for phosphorylase to mobilize glycogen in muscle.
This is so cool, I got it. Wow, it's so cool. Okay. So glucogen glycogen synthase is allosterically, sorry guys, I forgot how to speak. Glycogen synthase is allosterically activated by glucose 6-phosphate. I don't get that.
No worries. You're killing it. Let's clarify glycogen synthase and its allosteric activation. Glycogen synthase, which builds glycogen by adding glucose units from UDP glucose, is allosterically activated by glucose 6-phosphate. This means glucose 6-phosphate binds to a site on the enzyme, not the active site, tweaking its shape to make it more active. Why glucose 6-phosphate? It's a sign of high glucose availability, like after a meal, signaling the cell to store excess glucose as glycogen. This ramps up glycogen synthesis in liver and muscle when sugar levels are high. Clear? Want more on synthase regulation?
ATP inhibits glycogen breakdown. AMP activates more breakdown. And then G6P, so glycogen synthesis. So ATP and G6P kind of have similar effects there.
You're on the right track. Let's clear it up. ATP and glucose 6-phosphate, G6P, both signal energy abundance but affect different processes in glycogen metabolism. ATP inhibits glycogen breakdown by allosterically binding glycogen phosphorylase, slowing its activity, as high ATP means the cell doesn't need to break down glycogen for energy. G6P, on the other hand, allosterically activates glycogen synthase, boosting glycogen synthesis when glucose is plentiful, like after eating. Meanwhile, AMP activates glycogen breakdown by binding phosphorylase, signaling low energy and the need for glucose release. So ATP stops breakdown, G6P promotes building, both reflecting high energy, while AMP pushes breakdown for low energy. Okay. Clear?
Yes, that is clear. That makes sense. So can we talk also about regulation of glycogen metabolism by covalent modification?
Absolutely. Let's dive into regulation of glycogen metabolism by covalent modification, keeping it concise. Covalent modification means adding or removing a phosphate group to enzymes, switching their activity. Glycogen phosphorylase breaks glycogen, and glycogen synthase builds glycogen, are regulated this way. In low glucose, glucagon or epinephrine triggers a cascade. They activate protein kinase A, which phosphorylase kinase, which then phosphorylase glycogen phosphorylase, turning it into active phosphorylase A for glycogen breakdown. It also phosphorylase glycogen synthase, making it inactive, synthase B. High glucose triggers insulin, activating protein phosphatase 1, which removes phosphates, deactivating phosphorylase to phosphorylase B and activating synthase to synthase A, favoring glycogen synthesis. This phosphatization flip-flop ensures glycogen metabolism matches energy needs.
Okay, so let's also talk a bit about induction of CMP cascade and its opposite effects from glycogen synthase.
All right, let's cover the CMP cascade's role in glycogen metabolism and its opposite effects on glycogen synthase, keeping it tight. The CMP, cyclic AMP cascade, is triggered by low glucose when glucagon, liver, or epinephrine muscle binds receptors, activating adenine cyclase to produce CMP. CMP activates protein kinase A, PKA, which phosphorylase two key enzymes. One, phosphorylase kinase, which then phosphorylase glycogen phosphorylase to active phosphorylase A, ramping up glycogen breakdown to release glucose 1-phosphate. 2. Glycogen synthase, which gets phosphorylase and switches to inactive synthase
So,
B.
protein phosphatase goes from glycogen synthase B to A. Protein kinase goes from A to B. And B is less active, A is activated.
You're close. Let's clarify the roles of protein phosphatase and protein kinase in glycogen synthase regulation, keeping it concise. Glycogen synthase has two forms: synthase A, dephosphorylated, active, and synthase B, phosphorylated, less active. Protein kinase A, PKA, activated by the CMP cascade via glucagon, epinephrine, phosphorylase synthase A, converting it to synthase B, which shuts down glycogen synthesis during low glucose. Protein phosphatase 1, activated by insulin during high glucose, removes the phosphate from synthase B, turning it back to synthase A, activating glycogen synthesis. So, PKA,
That's okay.
H.
Okay. Thank you for watching part 2, guys. I'm going to do a final part 3, where we'll talk more about the electron transport chain. Thanks for watching.